First of all, I would like to thank you for the opportunity to participate in this fantastic project. And I hope that my expertise in calcium imaging will be helpful to all the students and also to senior people. And if you have any question while I'm presenting or at the end, of course, but afterwards, if you want to email me and if you want to further discuss anything in particular, it will be, of course, a pleasure. So, in this talk, I will explore the critical role of calcium signaling in the cellular processes and how we study its dynamics. Since this ion is so important, it's also very important to understand how it is regulated and how can we change the calcium levels in the cells. So I will begin by exploring the principles of calcium signaling and why measuring calcium signaling concentrations is really important. I will introduce key tools for visualization of the calcium transients, like bioluminescent proteins, chemical calcium indicators, and the protein-based genetically encoded calcium indicators. I also talk a little bit about which type of loading we can use. So if we load just one cell or if we load a network of cells, and if we are using a chemical molecule, this loading, of course, is different if we are expressing a protein-based genetic encoded calcium indicator. I will also talk where can we measure these intracellular calcium levels. And finally, I will talk about how we do it in our lab, because that depends a little bit of the facilities that you have for the measuring of the calcium level. So, why seeing calcium? Okay, so calcium signaling is everywhere. And I think that this concept, it's clear for everyone because we know that calcium signaling is important for cell cycling, cell cycle, but is also very important for the heartbeats. And these calcium levels, I'm going to point them. So these calcium levels in the cytoplasm can be controlled by several proteins, receptors, and channels that are expressed in the plasma membrane, but they're also present in the membrane of the endoplasmic reticulum or in the membranes of mitochondria. Okay, so we have numerous proteins that control this calcium signaling in the cells. And indeed, when we look into the central nervous system, we know that calcium signaling is very important for the neurons, since it can shift from acting as a cation that modifies membrane potential to one that serves as a second messengers following a receptor activation. So there's numerous functions for this ion, okay? And more important is action occur over a wide time range. So it can go from milliseconds when we are thinking on a neurotransmitter release, or it can go to minutes to hours when we think on gene transcription. Okay. He also differs in their time, since we can have different timings, different subcellular localizations, and also different amplitudes on this calcium signal and calcium levels in the cells. So it's very important to see the calcium and to quantify the calcium. So we need ways to investigate these quantities of the calcium that we have inside the cells. And other thing that is very important, I already told you a little bit how to keep this calcium gradient. So we have a lot of influx pathways that will lead to an increase of the calcium concentration inside the cell, in the cytoplasm of the cell. But of course, in order to keep this concentration low, we also have a lot of calcium buffering proteins and also calcium extrusion from the cells. So transporters that are responsible for removing the calcium from the cytoplasm. So, and just you also to keep in mind that the intracellular concentration of calcium is very low, is really low. It's between 50 to 100 nanomolar, while the extracellular concentration of this ion, or its concentration in the endoplasmic reticulum, is around 1 millimolar. And it's really important to keep these calcium concentrations low, because otherwise, if we have an increase in these concentrations, we will have activation of proteins that will damage the cell, and ultimately they can lead to cell death. Okay, so why is calcium so important in the nervous system, in the central nervous system? So first, we can think in the synapse. And we know that in the synapse, the quantity of the entry of calcium to the presynaptic terminal, it is very important because it's this entry of calcium through voltage-gated calcium channels upon the arrival of an action potential to the presynaptic terminal that will lead to the fusion of the synaptic vesicles with the with the presynaptic membrane, and in this way we have the release of this synaptic vessels content. And in this way we release neurotransmitters that will activate specific receptors, mainly in the postsynaptic neuron. And here I just have a representation of the receptors that allow the entry of sodium, but there are a lot of receptors that also allow the entry of calcium in the postsynaptic terminal, and in this way activates several signaling cascades that will have a specific effect. So calcium for the synapse is very important presynaptically, but also postsynaptically. In the recent years, it has been proposed the tripartite synapse. So it was considered that the presynaptic terminal does not communicate only with the postsynaptic terminal. There is a third component, which are the astrocytes. And when the neurotransmitters are released, they can also activate specific receptors that are expressed in the membrane of astrocytes. And the activation of these receptors in the astrocytes may lead to a calcium rise in this cell, as it's also represented here in this graph. And in some circumstances, this calcium increase in the astrocytes may lead to the release of molecules that are known as gliotransmitters that will modulate synaptic plasticity and synaptic transmission. Okay, so calcium has a very important role in the synapse, in the more classical synapse, but it also has a very important role in glial cells, such as astrocytes. So it's really important. This is an overview of the calcium, a very, very broad overview of the calcium importance for the central nervous system, not only in neurons, but also in glial cells. Okay, so we need to find, at a certain point, we need to find a way to quantify this calcium signaling in the neuronal cells and the glial cells. And for that, several calcium indicators were developed, namely the bioluminescent proteins, the chemical calcium indicators, and the protein-based genetically encoded calcium indicators, the GICs. And I will talk a little bit of all this type of proteins, and I will show you that some of them probably work better depending on the system that we have, and of course depending on the question that we are doing. So the bioluminescent proteins. Here is the example of a bioluminescent protein, iaquarin. Okay, she's derived from a marine organisms. And what happened with this type of proteins is that when the calcium binds to these proteins, it leads to the oxygenation of this group here, represented by the C. And what happens is that this group will leave this molecule, okay? And when there is this this removal of this group from this protein, there is the emission of a specific wavelength. So, what does this in what does this translate? It translates that when we have an increase in calcium in the specific system, the calcium will bind to the protein, we will have the release of this group, and we will have emission of light. Of course, that this type of calcium indicator has an advantage, which is we do not require external illumination of the sample, so we avoid phototoxicity, bleaching, autofluorescence, and then the undesirable stimulation of photobiological process. So all this part is very important because when we are using fluorescence to see a specific molecule or this to control these dyes, we always have this type of disadvantage: phototoxicity, photobleaching, etc., etc. However, this type of proteins have some disadvantage. So each molecule just performs only one emission cycle. Okay, so we cannot go back and have another emission cycle. Other big problem is that this protein cannot penetrate the plasma membrane, so it needs a gene transfection to be expressed in the cells where we want to evaluate this calcium signaling, and it has a low quantum yield and a low protein stability. So indeed, although they exist, they are not very used to evaluate this calcium signaling in cells. So later on, chemical calcium indicators were developed, and indeed some people that developed these indicators, they received the Nobel Prize of Chemistry. And basically these molecules, they are very interesting because they can cross the blood brain, they can cross the membranes of the cells, because they are a calcium chelator, which is this part here, coupled to a fluorophore, okay, based on a calcium selective chelator, okay. So they are conjugated with AM esters or dextran conjugated that will allow them to cross the membranes and to enter in the cells. Okay, they have different calcium affinities, which is also interesting because we can play depending on the type of cell and if we have more or less calcium in the cytoplasm to be evaluated. They have this emission and this excitation and emission spectra. They are pH sensitive. They are, of course, invasive because they will enter in the cell, and they do not allow for targeting to specific neuronal populations. So when we use this type of indicators, they will enter in neurons, okay, and they also will enter in glial cells, astrocytes, microglia, oligodendrocytes. So if we want to target only neurons or only astrocytes, this type of indicators, they are not a good strategy. And what does happen in this type of indicators? So when the calcium binds to this molecule, there is a slight conformational change in the molecule, and we can increase the emission of radiation when the calcium binds to these molecules. But there are two, this is very interesting molecules because there are two types. We can split the chemical calcium indicators in two types of dyes: the ratiometric dyes and the non-ratiometric dyes. So the ratiometric dyes, there is a ratio between two fluorescence intensity. This is a very, very interesting approach because this allows correction of artifacts due to the bleaching, changes in the focus, variation in the laser intensity, but makes measurements and data processing more complicated. Okay, and then we have the non-ratiometric dyes, in which on the binding to the calcium, the indicated used for the non-ratiometric measurement show a shift in their fluorescence intensity. But I will show you some pictures that will allow to understand this very, very properly. So this one is non-ratiometric dye. what happens here? So when the dye is free, so it means that we do not have a large quantity of calcium in the cytoplasm, the excitation will be low and the emission will also be low. What happens when we have an increase in the calcium, an increase in the levels of calcium in the cytoplasm? So this calcium will bind to the indicator, and we will have an increase in the excitation, but also on the emission. Okay, so we will have a direct correlation in the intensity of the fluorescence correlating with the calcium levels in that type of cells. As I said before, so the advantage is that we have an increase in the fluorescence signal, can be directly related to an increase in the calcium concentration. The disadvantage is that the fluorescence intensity depends on many factors not related to calcium concentration. Some of these are acquisition conditions, probe concentration, optical path, and other stuff that cannot be ignored, since it's a direct correlation between the fluorescence and, we think, the concentration of calcium. So then we have this ratiometric dyes. Okay, so we have two different types of ratiometric dyes. We may have one wavelength that will excitate the dye when she's free of calcium or bound to calcium. And then, depending if the dye is free, we will have a specific emission wavelength, or if the dye is bound to calcium, we will have another emission wavelength. Or we have some dyes that can be excited with different wavelengths, depending if they are binding to calcium or if they are not bound to calcium. Okay, so in this case, if the dye is unbound, is bound to calcium, it will be excited with this wavelength, and if it's bound to calcium, and if it's not bound to calcium, it will be excited with this wavelength. So the big advantage is that the calcium concentration estimated by these ratiometric compounds is not affected by the probe loading, the bleaching, the optical path length, the illumination intensity, because since we do the ratio afterwards in the analysis, we do the ratio between the emission, okay, we will cancel the bleaching, the problems in optical path length, illumination intensity. Okay, there is also some disadvantages. The acquisition, the data manipulation, it's more complex due to the use of fluorescence ratio. For instance, I'll talk then about the acquisition. You really need a system that will allow an acquisition between two wavelengths, for instance. Okay, and not all microscopes are suitable for the measurements with ratiometric indicators. And I give you an example of a ratiometric indicator, which is the one that I use in the lab. Okay, so just you have to know about this calcium indicator, which is the Fura 2AM. Okay, and the advantage of the Fura 2AM is that it is excited at two wavelengths, the 340 and the 380. Okay, and the ratio of the emission, okay, it's around 510, will give us a direct correlation with the intracellular amount of calcium. Okay, and as I said before, what happens with this dye? So the dye is stimulated at 340 when we have an increase in the calcium in the cytoplasm, and is stimulated at 380 when we have a decrease in this calcium concentration. Okay, this slide is just to give you an idea that there is several calcium indicators. On the left side, you have the non-ratiometric one. Some of them are very common. The fluorophore is very common to study calcium signaling in astrocytes. And there's also a lot of them ratiometric dyes, and Fura2 is here included. A very, very important thing, it's the affinity of this of calcium to these dyes. And as you can see, there are some differences in this affinity of calcium for these dyes. And this is very important. Why? Because when we are incubating our cells with a dye, this dye will work as a buffer of calcium. Okay, so in the cytoplasm, I didn't talk a lot about that, but we have proteins that work like buffers in the cytoplasm for calcium in order to keep the balance and the low concentration of this ion. So what we have to think is that when we add one of these dyes to the cells, we have to think that the calcium that is released for the cytoplasm will bind to the endogenous calcium proteins, okay? But it will also bind to these dyes that will work as buffers, okay? So we need to have a compromise of a dye that does not have a very high affinity, because if it has a very high affinity, it will be difficult to make sure that the calcium concentration that we are measuring is really corresponding to the reality, and it's not unmasked due to the fact that all the calcium is bind to the dye. So when we choose the dye, we also need to take this in consideration. Okay. So the two first dyes are I already talked about them. And finally, we have these protein-based genetically encoded calcium indicators. Okay, so we have a genetic approach. Okay, and we have this type of proteins that are able to sense calcium and also to be activated by a specific wavelength, and also to emit a specific wavelength that we can quantify the quantity of calcium in the cells. So we have two different types of GICs, the single fluorophore and the fret GICs. So in the first one, they have in his structure calmodulin, which is a protein that endogenously binds calcium. Okay, and we have two different types of proteins that can have this calmodulin, which are the kangaroo and the GCAMP proteins. I will be mainly focused on this one because this one are largely used in neuroscience field. Okay, and then we also have this FRET, the force resonance energy transfer based GICs, which also have calmodulin in their structure, and they also have this tropomyosin C in this structure. So there are different type of proteins that are involved in this FRET-based protein based genetically encoded calcium indicators. Okay, and I'm going to focus on the GCAM protein and also on the yellow chameleon. Okay, and once again, just to give you an idea, there are different types of GCAMP kinases. Okay, some of them are how do we choose a CAMP kinase? Some of them are faster than others to measure this calcium signaling, and if we think in this GCAMP kinase, GCAMP for an astrocyte, it can be slower. But if we want to use GCAMP in the neuron, it's better to be a faster one because the calcium signaling in neurons is faster. So we have to think on the type of GCAMP that we are going to use to decide what type we will choose. We also have different types of yellow chameleon and also different types of the troponin C based proteins. So all of them can be used to measure the calcium levels in different systems. As you can see, some of them, for instance, this fast GCMP are not so recommended for the mammalian system in vivo. Some of them do not detect spontaneous activity, and some of them do not detect the single spike detection. So we need to have all these variables into account when we choose the calcium indicator to our experiments. So how does the FRET-based GICs work? So the yellow chameleon. So what happens here is that we have this chimeric protein, which is composed of a blue or cyan variant of the CFP, which is the FRET donor. Then we have calmodulin, glycyl glycine linker, and then the KMB binding domain of myosin light chain kinase, the HAM13, and then a green or yellow version of the GFAP. So what happens is that these two, the CFP and the YFP, they need to be very close to each other in order to have a FRET signal. So we will excite the first protein, the CFP, at 440 nanometers. This protein will emit a photon that will stimulate the FRET acceptor if they are close each other, because if they are not close enough, this Venus will not be excited, okay? And we will not have an emission photon that will be recorded by our system, okay? So what happens is that when we do not have a high concentration of calcium, the system is not close enough. We do not have a FRET signal, but if we have an increase in calcium, there is a restructure, there is this conformational change in these proteins, and the CFAP emission photon can activate the yellow version, and we will have an emission. Okay, so this is how it works. Here is a schematic representation of what I just said. In this case, we have, in this situation here, and in this situation in yellow here, okay, we have no calcium. So the chameleon adopts this more flexible conformation, where the CFP and the YFP are completely separated. And in this conformation, there is a very low FRET probability. And indeed, it's represented here. We may have some signaling in the FRET signal, but it's not a very strong signal. When we have calcium in the medium, so there is this rearrangement of the calmodulin, and this conformational change makes that the CFP and the YFP, they come together. So when we excite the donor, we will emit a photon from the donor that will activate the YFP, and in this way we will have a FRET signal. And basically here is the difference, this dynamic range, that is the difference between the absence of calcium compared to the presence of calcium. And as you can see here, this is also some representative images of the increase of the calcium in the system, okay, the change that we will have in the fluorescence of this stimulated sample. Okay. And then we have the single fluorophore GICs, which are also known as the GCAMP family. So in this case, we just have a fluorescent protein, which is in this case the GFP, okay? And the GFP bind the calmodulin and the HAM13, okay? This protein is stimulated with this wavelength, so the 485 nanometers. And when the calcium binds to this protein, there is some change, some conformational intermolecular change that leads to an increase in the emitted fluorescence at 515 nanometers. So this is a direct measurement of the calcium increase that we have inside the cells. Okay. So what are the advantages of this type of calcium indicator? So they are applicable to nearly all organisms. Okay, so we don't need this ester permeation or hydrolysis, as I said in the Fura 2AM, for instance. Okay, they can be targeted to specific tissues, to specific cells. This is very important. So we can target neurons with this type of calcium indicators, or we can target glial cells with these calcium indicators. Unlikely to diffuse well enough to blur spatial gradients. Okay, good optical properties, visible excitation, emission rationing, high photostability. They are cheap. The DNAs and improved sequences usually are cheap to replicate and distribute. Disadvantages. So gene transfection is required. So if we are starting a new set of experiments, we will need to optimize in that specific brain area the concentration of virus that we'll probably need to inject to have a good expression in the area that we want of our dye. The maximum changes in the emission ratio is currently less than the small molecule dyes. That is also a disadvantage because usually the small molecules, they work, the emission ratio is really, really good. The binding kinetics are somehow slower, okay, than the chemical indicators. And the kinase and M13 might have some additional biological activity because they also exist endogenously. Okay, so until now we talked about the importance of calcium. We talk about the main calcium indicators that we can use to evaluate the calcium signaling in the cells. Okay. And now I would like to talk a little bit about how can we load our cells or our system with these calcium indicators, okay, into neurons, of course, or into glial cells. So there are different types of dyeing loading approaches. We can have the single cell loading, the acute network loading, or the expression of the GIS. Okay, so here is a schematic representation of these different types of loading. Okay, and most importantly, of course, we will do the loading depending, of course, of our specific scientific question. So how can we load one single cell? For that, we just have to do three different things. Or we do a sharp electrode, or we do whole cell patch clamp, or we do single cell electroporation. In these three techniques, it's implicated that an electrode, a very thin electrode, it's filled with our dye, the one that we want to introduce in the cell of interest. And then, of course, we have to, in this case, to perforate the cell and to load the cell with this calcium indicator, or I have to do a whole cell patch clamp, and the cell and the dye will diffuse from the pipette into the cell of interest, or we can also have this electroporation of the cell, so we destabilize the membrane and the dye will enter in the cell. So this is you have to think that this is a very specific, if we want a very specific assay. So imagine you want to record the calcium signaling in only one neuron, for instance, to see if the different parts of the neuron respond differently in terms of calcium, respond differently to the stimuli, or you can also apply the same way of thinking to an astrocyte. So we have to do this type of loading of the buffer. And then we have this bulk loading of multiple cells. Okay, we can use the AM esters, the dextran conjugated, and both provide acute loading. So what does this mean? It means that we can go to a specific area of the brain. Okay, for instance, this is the cortical area. We will have this pipette, and the dye will diffuse in a specific area of this specific region. Okay. Here we will, of course, we will load several cells that are in that specific brain area that we want to analyze. Okay, and we will look into all these cells that load the dye. Another thing, we can also load a specific uh, a specific um, um, group of fibers. And in this case, by loading a specific group of fibers, the neurons that project, the projection of these fibers will be loaded with the molecule to measure the calcium signaling. And we can also do this electroporation to administer, okay, because here there is this AM loading, because the AM group can cross the cell membrane. Also, the dextran conjugated can cross the cell membranes. But we may have some calcium indicators that do not have these groups. So the electroporation of the tissue will allow the entry of the dye inside the cells. Okay, and finally, we have the expression of the proteins, and this will usually is done by stereotaxical viral transduction. Okay, transgenic technology. So there are some strains of mice that already have specific types of cells that also express a CAM kinase in their specific cells to evaluate this calcium signaling, or the combination of two approaches. So imagine that you already have a transgenic mice that express a specific protein, and you will do a viral transduction to express a second protein. And here is the same thing. How we will inject the virus in the specific brain area that we want to evaluate. Okay, we can do this in utero evaporation, and will the pups will have this expression in their cells, or we can, of course, produce transgenic mice with the inclusion of these proteins that will be used to quantify the calcium signaling. So we talked about the calcium dyes. We talked about the ways that we can mark the cells, the ways that we can have these calcium indicators inside the cells to evaluate the calcium. And of course, we have to observe this fluorescence, okay? So we can use normal fluorescence microscopes. We can use confocal microscope, but there's no need of having a confocal microscope to do this calcium imaging, okay? We can do it with a normal fluorescence, epifluorescence microscope. But of course, if we want a more, with more definition, we can use a confocal microscope. We can also use the two-photon microscope to have a big resolution of specific processes in the neurons or in the astrocytes or in microglia, for instance. And we can do this will be ex vivo. Usually the confocal is also ex vivo. They are the two-photon we can do in vivo with head restraint of the mice, the animal. We can do in vivo using the two-photon. And we also have this endoscope, which allows to place an optical probe inside the animal's head to measure this calcium signaling. So there is this insertion of optical fibers in the endoscope, and we also have these portable microscopes with that fixed microscope that will allow the recording of the calcium signaling while the animals freely move. So there are a lot of ways to do this recording. So ex vivo, in vitro, in vivo, and there, of course, that the complexity of these different approaches, it's very very different, but indeed we can do all this type of recordings. And finally, so we can measure where can we measure this intracellular calcium signaling? So we can measure in the dendrites and spines, okay, in vitro and in vivo. We can imaging post and presynaptic and postsynaptic terminals, okay, the function of these terminals, and we can also measure astrocytes in vitro and in vivo. Okay, we can do circuit analysis in different animal models in vivo, and we can also do calcium imaging in behaving animals. So we can do completely different things with this technique, and we can go from very tiny processes and specific areas of a neuron or an astrocyte, and we can imaging the not the whole brain, but a specific area of the brain, while the animal is performing a specific task. And I'm just going to give you some examples. In the all slides, you have the paper, so you can go there and check. But here is there to show you that it's possible to imaging the post and the presynaptic function in vitro. So here we have a postsynaptic CA1 pyramidal neuron, and we have these specific regions in the neuron that are represented here, and we can see this calcium signaling in these neurons. Here we also have this small area that is amplified. It's a Purkinje cell, dendrite cell. These numbers are also identified here, and as you can see, we can also see the calcium signaling in these areas. Okay, and here we have this presynaptic bottom in the cortical layer 2-3, and we can also see the calcium signaling when we have this change in the membrane potential. So we can do indeed this very specific observation of the presynaptic and the postsynaptic terminals. We can also observe the dendritic and the spine calcium signalings in vivo. So here I have two examples. In this one is the dendritic calcium signaling from the layer 2-3. It's a pyramidal neuron, okay, in the vibrissae cortex. And in the top, you have the calcium signaling, and in the bottom, you have the electrical signaling. And it's shown here that we can measure this at different dendritic spots of the neuron. Here we have a spine calcium signaling for a pyramidal neuron. And as you can see, we have this definition. So as you can see, this is the pipette. So they did patch clamp with this dye. So there was the dye spread through all the neuron. And then they record this portion here that is amplified here, and they mark different regions. And here you have the correlation of these regions to these traces. And you can see, so this is an in vivo experiment, you can see when there is a sound to these animals, you can see that the different parts of this pyramidal neuron have different responses to the sound. Okay, so this calcium signaling. And we can also see astrocytes in vitro. So it's the same procedure. In this case, it's a GCAM-P6 protein. Different spots of the astrocyte were selected. and it was observed the calcium signaling in these different spots. Okay, and also this is a calcium imaging in the behavior animal. So, and this is just to give an idea that it's possible to monitor the calcium signal in vivo while the animal is performing, in this case, behavioral protocols regarding the choice of an olfactory related choice. And as you can see, we can then monitor how this net of astrocytes probably correlate or not. We can select different astrocytes and to see what happens in these cells. So finally, how do we do it in the lab? Okay, and now I'm going to talk specifically in our lab. Okay, so in our lab we have already made calcium signaling experiments in different types of cells, but we always use primary cultures or cell cultures. I don't have cell cultures here, but we also already did it. We already did experiments in primary cultures of astrocytes, primary cultures of neurons, the SVZ-derived cells, dorsal forebrain organoids, superior cervical ganglion neurons. And we have, what is the problem of our system? So we have in the lab an inverted epifluorescent microscope. So what happens in our lab is that since we have this type of microscope, we just can observe uh, the calcium signaling in cells that are on a monolayer in the plate. Okay, because if we have not an inverted one, but a microscope that have the objectives on the top, we could also do it in slices because we could have a distance of work that would allow us to go a little bit inside the slice, and we could also do this in slices. But since we have this specific equipment, we are kind of limited to perform our calcium signaling experiments only in cells that are in a monolayer in a specific plate. And because of that, we work with primary cultures or cell lines. Okay, so usually we can do, it depends the cells that we want to study. We also use, usually in the lab, the FURA 2AM. Okay, so the FURA 2AM, as I tell you before, is a ratiometric dye. So we can stimulate FURA 2AM with two different wavelengths, and we record at one wavelength. Okay, so we incubate our cells with this dye at a concentration of 5 micromolar for 45 minutes at room temperature in a HEP solution. Okay, we always use plates with several chambers or a petri dish, but they need to have a cover, a glass cover slip on the bottom due to the objectives that we use. So usually they are not on plastic, they're on glass, and that's important. And this is usually the solution composition that we use in our lab. And it's in this solution that the cells are incubated with the FURA 2AM, and usually is this solution that we use to perform the calcium signaling recordings. So the FURA 2AM, what happens is that we have the molecule outside the cells. Since, as I already explained you, the FURA 2AM has these AM groups, is able to cross the cell membranes, and once the FURA 2AM is inside the cells, this acetoxymethyl groups of the FURA 2AM are removed by cell sterases, and the FURA will be trapped inside the cell. So the FURA cannot go outside the cell anymore, and we will have these cells loaded with a dye that we will use to measure the calcium concentration. Okay, And since it's a ratiomeric dye, what happens is that when we have an increase of calcium in the intracellular space, in the cytoplasm of the cells, we will have an increase in the fluorescence that corresponds to the stimulation of 340, and we will have a decrease in the fluorescence that corresponds to the 380. So this means that there is a calcium increase that binds to the dye, and this one means that there is a calcium, there is a signal decrease in the dye that is not binded to calcium. When you do the ratio, of course, because it's the 340 by 380, we will have an increase in this ratio. I also mentioned in the beginning that the problem of this ratiometric rise is, of course, the analysis of the data. So we use in the lab, we use the MetaFluor software that already do this ratio between one wavelength and the other wavelength. So we do not have to do additional calculations to do this ratio, and we will just analyze it, as I will explain you, the frequency of the transients, the amplitude of the transients, that are one of the main goals of our analysis. Okay, so here is the system. So after the incubation of the cells, the glass bottom dishes, they are mounted in our inverted epifluorescence microscope that is equipped with a Shannon lamp and this bypass filter of 300 and 314 and 380, which is the lambda DG4. Okay, so this is a very important component in this type of ratiometric recordings, because what does this apparatus allows is the high-speed shutter function that opens and closes times at of 500 microseconds. So this means that we can switch from one wavelength to the other one very fast. Okay, and this is important. Okay, because usually this can be changed depending on the type of cells that we have. Can be this excitation time can be changed, but usually we excite for 200 milliseconds with 340 and 200 milliseconds for the 380, and then we do the ratio between the emission of these two wavelengths. So if we do not have this apparatus, which is very fast, we will not able to catch all the calcium oscillations that we have in our cells. Sometimes it's more difficult to optimize in the confocal because you really need a confocal that do this acquisition very fast. Otherwise, you will lose some calcium transients, some calcium oscillations in your samples. And indeed, this type of machine is an advantage for this type of experiments. So we set the setup, we plate the cells, we turn on everything, and then we start the acquisition of our frames. Okay, and here is an example of one of our samples that we get. Okay, so what do we have if I go back and if you look just here to the screens of the computer, you can see here 3 different windows. So one of them corresponds to the 340, the other one to the 380, and the other one to the ratio between these two wavelengths, which is represented here. Okay, so after obtaining, and this is a sequence of images, it's like a video. Okay, I thought the previous one was a video, but it's not working. So after that, we need to go and select the cells of interest. Okay, in this case, I'm not using a confocal. Usually I evaluate the calcium signaling in the sum of the cells because I do not have the resolution, okay, to measure the calcium signaling in the process of the cells. So here in the lab, we usually measure the calcium signaling in the soma of the cell. So I select the cells of interest, so this ROIs, the ROIs of interest, and this is correspond to the 340, 380, and here the ratio. And afterwards, the program allow me, okay, to construct this type of graphs, having in mind the response a long time that the cells have to a specific stimuli, okay? And the software also allow me to export these results as an Excel file that will be analyzed afterwards, okay? So here I have some examples of our experiment. So I have this path incubation. I don't know, should work because there are some videos, but they are not working. This was the baseline before the application of GABA. And this was the bath incubation. This is very important because here we can start changing the protocols. Okay? We can do a bath incubation of a specific drug. In this case, I was interested to see the calcium signaling of this type of astrocytes to a GABA incubation. Okay, so I incubate all the cells in the plate with GABA, and I measure the calcium activity in these cells. Okay, and it was observed an increase in these calcium oscillations when I incubate the cells with GABA. We measured the frequency of the transients as well as the amplitude of these transients. What can we also do? We can do a proof application of a specific drug. So in this work, we give a proof, of ACEA, which is an agonist for the CB1 receptor. This proof will be concentrated only near some cells. So here is the results that we obtain. Okay, and then we treat the cells with different agonists and antagonists. In the first one, we did not treat the cells with anything. So we have the effect of CB1 receptor activation, and there is this increase in calcium, as you can see by the change of the color of the astrocyte. And then we treat the cells with CGS, and we lose this effect of the CB1 receptor. So, but here, instead of a bath application, we just give a puff to a specific cell. So it's much more controlled than to have this bath application of a specific molecule. Okay. And then we also have a very interesting work with the Professor Sara Chapelli, in which we incubate the cells with different molecules that will give us an indication if the cells are more compromised to be neurons or if they are not still compromised yet to be neurons. They are more glia-like. Okay, so in this study, if you want to check how we do that. So we have this 5 minutes of baseline. After the 5 minutes of baseline, which correspond to this part here and this part here, we incubate the cells with potassium chloride. So potassium chloride will depolarize the ones that are already neurons, okay, for 2 minutes. Then we wash out the cells, and then we incubate them with histamine that will mainly activate cells that are non-neuronal cells yet, okay? So by doing the ratio between the quantity of cells that responds to potassium and the ones to respond to histamine, we may say that we have a larger or lower percentage of neuronal-like cells or these immature like cells that are not still yet compromised of being neurons, okay? All this is also, all this type of experiments, it's made through bath incubation of the cells with potassium or with stamine. Okay? So, taking home message, why do we want to observe this calcium signaling? As I said in the first slide, because calcium signaling is everywhere and it controls, the main focus of this seminar is to talk about the methodology. But of course, it's this methodology. It's very important because if calcium signaling and calcium is so important and is everywhere, it's important to understand how it works, how it's controlled, what is controlling it. So it's very important to study calcium signaling cells. And then I explain a little bit how can we measure this intracellular calcium concentration. I talked about the tools that we have to measure calcium, the bioluminescent proteins, the chemical calcium indicators, the genetic approach, the way we load the cells. Okay, that is also very important if we just want to look into a neuron or to an astrocyte, if we want to look into a network of cells. Okay, that's also a very important question that we need to have in mind when planning an experiment. Okay, of course, the type of devices that we use to visualize this calcium signaling. And then I talk about where can we measure intracellular calcium signaling. We basically can measure everywhere. It depends a little bit, of course, of the type of the tool that we are using to analyze the calcium signaling, the way we load the cells, and the imaging device that we have. And then I explain you a little bit of the things that we have in the lab and how we use that things in our lab. So this what I have prepared for you today. And now I'm open to questions or anything that you would like to discuss with me. Thank you very much.